Good morning, everyone. Today we have Dr. Alan Goodman from Washington State University here with us on Science Mission. And I want to give a little bit background about him first. He completed his PhD from Washington University, Seattle in bioengineering. And because of his passion for working in science, later on he continued various postdocs. He worked as a postdoc at Washington University, Seattle. He moved to Spain to continue his research. Then he came back to United States and he worked in University of Miami, Florida. And later on, he accepted a position as assistant professor at Washington State University, Pullman, where he is currently working. He is a passionate researcher, and he has worked in different areas, including virology, immunogenicity, as well as host-pathogen interaction. So throughout his career, he has mentored many students. He has published 20 plus publications. He is an active member of various societies. He has worked as a reviewer as well as editor for various publications. And apart from that, he has also served as a mentor as well as consultant for some of the biopharma biotechnology companies. So I would like to welcome Dr. Allen and ask him some questions. Good morning, Dr. Allen. Nice to have you here.
Hi, thanks for having me.
Can you hear me?
Yes. Are you able to hear me?
Yeah. Give me 1 second.
Okay.
Good morning, Dr. Allen.
Hi, good morning. How are you?
So I would like to ask you a few questions. Would you like to share some of your background?
Okay, good.
So about your research and what motivated you to continue to work in this field?
It really all started when I was an undergraduate at Johns Hopkins and working in various labs, first in a lab of infectious disease, and then chemical engineering, which was really more like a bioengineering lab, and working on a project where we would trace how beads, fluorescent beads, moved throughout a cell and moved throughout the nucleus. And we hypothesized that these little beads could almost act like, their movement could be like how a virus moves through a cell. So thinking about that and applying it, how these motions of these beads move and thinking about them as viruses, spawned me into, wanting to study virology. So that's what I did at University of Washington and pretty much throughout my scientific career. Now I can't hear you anymore. No, still can't hear. Still nothing.
Oh, sorry about that. Thank you so much for the introduction. And would you like to share some information about how many students are there in your lab? What kind of projects are they doing, and what are some of the long-term goals?
Yeah, so we have right now 2 graduate students that are permanently in the lab. We've had a number of rotation students, so likely taking on another student starting next year. And there's also about 6 undergrads in the lab, anywhere from junior to senior level undergraduates at Washington State University, and a lab manager. So yeah, we have a good lab, good size, a lot of motivated students, all working on various projects that I'll talk about during my presentation, but looking at all related to the host immune response and using different bacterial and viral pathogens to understand ultimately disease. And we use a fly model, as I'll talk about, because we can transfer the knowledge gained in Drosophila to a mammalian system, especially when the host proteins or host genes that we're studying have similar function.
Great, yeah, great to hear that. And I know you're a busy person. You have so many graduate, undergraduate students to work with. Apart from science, what else do you enjoy doing? What are some of your hobbies?
Yeah, so spending time outside, spending time with the family, watching, playing sports, any kind of outdoor activities, reminiscing in old video games when I have a chance. So yeah, things like that, showing my 2.5-year-old son how to play very old Nintendo games and helping him learn to do that kind of stuff.
Nice, that's great. And also, is there a particular role model that you always look up to?
I'd say all of my past, mentors in the labs, they've all given me some perspective on how to be a good scientist, how to mentor other students, how to deal with the ups and downs of academic research. So I think I owe it to a lot of my future success to my past mentors.
That's great. And one last question, as this broadcast will be broadcasted for all the scientists out there, if you would like to share some kind of vision that you have or some kind of collaborations you're looking for that might be helpful, this might be a good chance to share that information.
Yeah, I think ultimately, like I said, we work in a fruit fly model system, looking at this very small organism where we know everything about all of its genes very well. So looking towards collaborations into a larger animal model. We're here at vet school, so we can work with these, apart from mammalian mouse models, we can look at large farm animals. Trying to move the research into that. Some of the viruses we study infect some of the animals that are here at our vet school. Looking for collaborations in that regard would be fruitful.
That's great. Yeah, with that, I would like to request you to share your presentation, and feel free to go ahead and start whenever you're ready.
Okay, thank you. Okay. So are you able to see the screen okay?
Yes, that's great. Yeah.
Okay, so again, thanks for having me. The title of the presentation today is Conservation of Host-Pathogen Interactions and Innate Immunity in Insects in Mammals. So I'll start by giving a very brief introduction to innate immunity. Maybe my cursor, is that able to be seen on the screen?
Yes. Okay,
good. So innate immunity is an ancient form of host defense initiated upon foreign pathogen infection. And the innate immune response is triggered when a cellular pattern recognition receptor, PRR, encounters a pathogen-associated molecular pattern, or a PAMP. Some very basic examples of this are, what happened there? There we go. Are the toll-like receptors 3, 7, and 8, RIG-I or PKR that can recognize motifs of a viral infection, or other toll-like receptors like 1, 2, 4, 5, 6, and 9 can recognize byproducts of bacterial infection. So sometimes when people think of innate immunity, they see this big spaghetti map of all these different receptors, different intracellular mediators, transcription factors, and it can look pretty daunting to try to understand how all of this plays together. But one thing about this map is that you can see there's some color coding. So we like to think of it as we think of the receptors, these intracellular mediators as families, and then these transcription factors here in blue. And then we specifically focus on just a couple of these pathways, some of which are, like I mentioned, the toll-like receptor signaling. So that's just one part of this really big map, or what's called the RIG-I-like receptor signaling, recognizing viral, in this case, RNA, double-stranded RNA, but also other nucleic acids that can be sensed during a viral infection. So then to break it down into much more simpler terms, as I mentioned, there's the pathogen-associated molecular pattern that is produced during viral or bacterial infections. And these act as the input to the pattern recognition receptor. So then once that pattern recognition receptor becomes activated, it'll signal through these adapter molecules and then activate transcription factors, which can translocate to the nucleus. In the innate immune response, one of the major outputs from the input of an infection is interferon beta. So then once interferon beta is induced, and it can be secreted from the initially infected cell, And interferon beta will engage the type 1 interferon receptor, which will activate, again, some other mediator molecules can activate the transcription factor STAT1, which will then translocate to the nucleus and cause the induction of these interferon-stimulated genes. So just through the induction of a single molecule like interferon beta, there can be the induction of hundreds of these interferon-stimulated genes, which will really activate the immune response, will allow for a bridge to ultimately an adaptive immune response, because these interferon-stimulated genes will recruit other cells to the site of infection that can then take up antigen and cause a further adaptive immune response. But we're really focused here on this initial set of signaling events from the input of the infection to looking at the output of that first transcription event. In this case can be interferon beta, and as we'll talk about in a fly model, there's things called antimicrobial peptides that can be induced during the innate immune response. We also focus on a molecule called STING, which is a nucleic acid sensor. It recognizes cyclic dinucleotides, and I'll focus a little more on that pathway in this diagram. So when STING gets activated, it translocates and can signal interferon beta induction. So let's look over the mechanism on how STING gets activated. So during viral or bacterial infection, there can be intracellular DNA, which will activate this molecule called cGAS, which stands for cyclic GAMP synthase. That'll cause the metabolism of cyclic dinucleotides, which then can directly bind to STING, which normally sits at the endoplasmic reticulum. When STING gets activated, it actually moves to areas that are perinuclear and will activate NF-kappa B or IRF3 transcription factors that will translocate to the nucleus and cause the induction of interferon beta. So the take-home message here is that intracellular DNA due to bacterial infections or viral infections will activate cGAS, which activates STING, which cause the induction of interferon beta. So an important part about this diagram is that in any innate immune pathways, that if there's defects in any of these pathways, an autoimmune disease can develop. So autoimmune disease is a major health burden, can affect 50 million, it affects 50 million Americans. It's the top 10 cause of death in women under the age of 65, the second highest cause of chronic illness, and results in a $50 to $70 billion burden annually. So some of the diseases that are caused by, or that some of these autoimmune diseases that are caused by defects in innate immunity are Crohn's disease or irritable bowel syndrome, lupus, Sjogren's disease, or Aicardi-Goutieres syndrome, which causes defects of innate immunity in the brain. There's also this disease called STING-associated vasculopathy, which there's an actual mutation in the molecule called STING. STING is hyperactive and causes these innate immune responses in the absence of any sort of infection. So innate immune dysfunction is central to these painful and costly pathologies. So we recently reviewed some of these defects in both the nucleic acid sensing, defects in nucleic acid sensing of double-stranded RNA, so defects in RIG-I signaling, or other molecules that are part of this RNA sensing pathway. So when there are defects in some of these genes, we get, there's a number of autoimmune diseases. And similarly, defects in DNA sensing pathways can cause autoimmune pathology. So again, I mentioned mutations in STING cause this SAVI disease, which because of the basically all these autoimmune diseases are hyperactive, hyperactivation of some of these nucleic acids sensing molecules. And so they're basically signaling in the absence of any infection and causing the induction of these pro-inflammatory cytokines, which result in autoimmune diseases. So the question that our lab asks is, can we utilize orthogonal animal models to better understand the innate immune pathways as they relate to autoimmunity and specifically to microbial infection? Because the overall idea is that any kind of mutation or any kind of activation of these genes, of these proteins, first off, in the absence of infection, it will result in autoimmunity. But if these proteins can't signal properly during infection, then we have this infection pathology. So some of our current lab projects are one in insect virus-mediated immune responses in mammalian hosts. We use Drosophila as a model for Coxiella burneti infection. We're characterizing the Drosophila homologue of sting, and then we're looking at the intersection between innate immunity and translational control during Zika infection. So again, I mentioned that we use the fruit fly model as a major model to understand how do the innate immune responses evolve and differ between this invertebrate and potentially these mammalian systems. So we're doing a lot of our work in the fruit fly model to ultimately make discoveries of novel genes that we can ask, do they function similarly in a mammalian system? And if so, how can we pursue looking at those genes, at those proteins during infection in a mouse model? But we're not quite here yet. We're still trying to do all of our basic research in the fly model. So some of you may know this, some may not, but innate immunity was actually discovered in fruit flies. So fruit flies live in a very caustic environment. There's always a number of bacterial or fungal infections that can occur. There's parasitic wasps that will attack the pupa of fruit flies. And then, and this is a fungal infection. So this picture is actually from a fruit fly that is lacking the toll protein. So similar to a mouse model that has the toll-like receptors, the toll gene was identified in fruit flies, and when mutated, it's overburdened by this fungal infection. This is what led to the Nobel Prize in 2011 to Jules Hoffman, who he shared with a couple other researchers who then further studied the toll-like receptors in mammalian models. So again, I mentioned that we want to utilize this orthogonal fruit fly model to extrapolate into the mammalian model and other insect models of pathogenic infections. So again, going back to the lab projects, we'll start by, I mentioned these 4 projects, but again, the whole goal is to uncover these evolutionarily conserved host factors that are critical to the host response to infection. And I'll start by talking about these 2 projects that are looking at insect virus-mediated immune responses in mammalian hosts, and then how to target the innate immune response to reduce West Nile virus infections. And these are primarily led by my graduate student, Laura Allers, and a couple of undergraduates, Chastity Trammell and Grace Carroll. And here's Laura, so we'll first start talking about this first project. So we used a DNA virus called Invertebrate iridescent virus 6. It's a double-strand DNA virus, but we found that it activates an RNA sensing pathway in insects. So just a little bit of background about Invertebrate iridescent virus 6. So in flies and mosquitoes, the RNA interference pathway is activated as an immune response to many viral infections. So RNAi acts as this, how RNAi acts as an immune response in mammals is still controversial. So IAV6, a very large double-strand DNA virus, 212 kilobase genome. The size of the virion, quite large, 200 nanometers in diameter. So we wanted to ask, how does the mammalian innate immune response react to this insect viral infection? So it does initiate an innate immune response in mammalian cells. And again, just looking at, back to the pathways that during an innate immune response in mammalian systems, there's activation of these transcription factors, IRF3 and NF-kappa B, that lead to the induction of type 1 interferon. So here we're showing that during IIV6 infection, we can get phosphorylation and activation of IRF3. And then we get secretion of interferon beta by 24 hours post-infection of post-IIV6 infection. So we also see NF-kappa B. So this is looking at translocation of NF-kappa B to the nucleus during IIV6 infection. So you can see very robust activation of NF-kappa B as marked by how it translocates to the nucleus. But interestingly, what we found is that while IIV6 will infect mammalian cells and cause an innate immune response, it actually does not replicate. So normally it replicates very well in Drosophila S2 cells. So you can see this, what's called a basically a viral factory. You can see all these virions that are produced, some of which contain nucleic acid. So the dark spots show that it's a fully infectious virion that contains the viral DNA. Some of these capsids are still open because they haven't packaged the DNA. But during infection in mouse embryonic fibroblasts, we can see these virions that have infected the cell, but they're not replicating. And we observed that there was no increase in IAV6 in mouse embryonic fibroblasts, both by using qPCR methods and TCID50, or the ability of infectious virions to be replicated. We next, as again I mentioned, that a lot of our goal is to use other tools to understand how Drosophila, how insect immunity and mammalian immunity are similar, and we found that IAV6 activates a conserved immune response in mammalian cells. So here is showing the dicer molecule, which is part of the RNAi pathway, and dicer in Drosophila contains this helicase domain, which is important for binding and sensing to double-strand RNA. And in there's very high similarity in this helicase domain to human dicer and actually human rig-I. Again, that's the major innate immune double-strand RNA sensor in the mammalian systems. So high similarity among these helicase domains between Drosophila and human species. So what we did here is look at, if we knock out some of these genes in a mammalian system that contain these helicase domains, how is the interferon beta secretion affected during IAV6 infection? So what we did was knock out dicer, rig-I, MDA5, or MAVs, and we saw that during infection in these knockout embryonic fibroblasts, during IAV6 infection, there's markedly reduced secretion of interferon beta. So we next asked, now that we made some, we discovered how IAV6 activates an immune response, but it doesn't replicate, we asked, could the IAV6 mediated immune response, so this induction of interferon beta, could it protect against other viral infections, especially types of infections that can be transmitted from insect vectors to humans? So we looked at how VSV or West Nile virus infection occurs following IAV6 infection. So what we did was we infected mouse embryonic fibroblasts with IAV6, and then we collected that supernatant of the infection and transferred them to new mouse embryonic fibroblasts. And then we infected with West Nile virus or VSV, and we measured West Nile virus. We used the Kunjun strain of West Nile or VSV by plaque assay. So we noticed that at different times post VSV infection, we would see a reduction in the level of VSV replication following priming of cells with IAV6, of supernatant from IAV6 infected cells. And same with the Kunjun virus or West Nile virus, we saw a marked reduction in the ability of West Nile virus to replicate after the cells were primed with IAV6. So overall, we found that the RNA-mediated immune response to IAV6 could protect against subsequent arboviral infections. And the next question we wanted to ask was, what genes drive the insect immune response to West Nile virus infection? So first, what we had to do was show that West Nile virus infection is lethal to adult Drosophila. So here we see, so either infected with West Nile or mock infected, we can see, we see mortality of these flies in wild-type flies, but a different wild-type fly strain, we observed that there was no difference, there was actually no lethality. But the key here is that these are two different types of wild-type flies literally caught from the wild. So line number 28 or line 566. And what these are strains of over 200 fly lines that are part of the Drosophila genetics reference panel. So this is a panel of 209 strains of wild-type caught flies caught at a farmer's market in Raleigh, North Carolina. And all of these 209 strains have their genome sequenced. So what we can do is we can perform infections in all 200 fly lines, look at the rate of mortality, and then perform what's called a genome-wide association study to identify genes that are controlling the rates of mortality in these fly lines. So the idea is to identify gene variants that are significantly associated with the complex trait of, in our case, response to West Nile virus infection. And again, I mentioned that these projects are carried out by Laura and also Chastity and Grace in the lab. So we infected our fly lines and observed a normal distribution of hazard ratios, which is essentially the ratio of mortality rates from mock-infected flies to West Nile virus-infected flies. And then we were able to perform the genome-wide association study using the hazard ratio as input for that algorithm. We then were able to produce a Manhattan plot showing some of these so genes that have high significance and are likely to somehow control infection. So the next steps are to do mechanistic studies in flies that are mutant for some of these genes. And we also were able to do gene set enrichment analysis. So taking all the data of every single p-value for every gene, we are able to input that into this gene set enrichment analysis and do gene ontology studies. And we found that many of the gene ontology categories that are significant could have some relation to viral infection. So again, looking at ATP, ADP transport activity, so looking at energy, activation of the JNK pathway, regulation of the toll pathway, again, an immune response. So RNA processing, so we mentioned earlier that RNA viruses activate RNAi as an immune response, so we're seeing genes, sets of genes that are significantly regulated that are part of some of these pathways. Dead box helicase, so again, these helicase domains that are part of sensing double-stranded RNA, we can see significant regulation of genes in these categories. So again, I mentioned the next steps are to mechanistically dissect the function of these genes during West Nile virus infection in fly and also in mosquito models. And as I mentioned, much earlier during the introduction, we ultimately want to take this into a mammalian model, potentially a horse model. So at WSU, at the vet campus, we actually get a number of horses here that have been infected with West Nile virus in the wild. So can we use some of these large animal models to look at West Nile virus infection? So moving on to the next set of projects, I'll next talk about the Drosophila models of Coxiella burnetii infection and how that relates to the Drosophila sting protein. So these experiments were primarily carried out by Zach Howard, an undergrad in the lab, and Aiwi Hirarasu, our lab manager. So to step back, so what is Coxiella burnetii? So it's a gram-negative bacteria. An infection with as little as one bacterium can cause disease in 50% of subjects. Coxiella mostly infects ruminant animals, so they're commonly infected and act as a reservoir for human infection. So farmers who come in contact with these animals, Coxiella causes spontaneous abortion. So if that's occurring in goats, sheep, cows, then farmers can become sick and local epidemics can occur. Wild-type Coxiella is a Category B bioterrorism agent, so any work with Coxiella burnetii requires BSL-3 facilities. So here's one of the major virulence factors of Coxiella burnetii is this long lipooligosaccharide from this O antigen that's on the surface of Coxiella burnetii. So this is the virulent phase 1 strain of Coxiella. But an avirulent strain was cloned and produced in 1986 at Rocky Mountain National Labs. And simply that avirulent phase 2 strain lacks that O antigen, which can subvert innate immune responses and subvert all immune responses in infected animals. So we wanted to ask, will that avirulent strain infect Drosophila? So this phase 2 strain we can work with in BSL-2 facilities. It does not infect mammals with competent immune systems, but any kind of knockout mouse that lacks toll-like receptors, this phase 2 strain is virulent. But we want to ask, will it infect and cause mortality in wild-type Drosophila? So we found that Coxiella infection is indeed lethal to adult flies. So looking at both males and females, infecting them with 10,000 bacteria per fly, we can see a drastic decrease in survival in both males and females. And we saw that that the bacteria is indeed replicating in these flies. So using what we're doing is measuring genome equivalents. So this is a way to measure how many essentially Coxiella genomes there are in infected animals. And the reason we do that is because doing colony forming units for Coxiella is much more difficult. So we can see that by 30 days post-infection, even if we infect flies with a low dose, 100 bacteria per fly, or a high dose, we can see marked increase in replication from 6 to 30 days. We can also use a polyclonal antibody to recognize Coxiella antigens in infected flies. So we can see these Coxiella antigens are the same ones that are recognized in just the infection inoculum. And then we can see that there's an innate immune response in the fruit flies as marked by the induction of this antimicrobial peptide called drosophila. So in infected flies at 12 days post-infection, we get activation of this antimicrobial peptide that's primarily activated by an immune pathway in flies that mediates gram-negative bacteria infection. So when you look at a pathway that's induced during a gram-positive bacterium, we don't see induction of this molecule called defensin in these infected flies. So again, only the pathway that's activated by a gram-negative bacteria get activated, and again, Coxiella is a gram-negative bacteria. So what cells does Coxiella infect in the fly? So normally in mammalian systems, Coxiella infects monocytes circulating in the blood. So we wanted to ask, well, the equivalent to blood cells in the fly is called a hemocyte, and we asked, are the hemocytes getting infected? So what we did is we used Drosophila larvae and put them on a plate of Coxiella. Let them. We would prick the fly so that the bacteria can invade and infect the larva. And then we would dissect out these larvae to extract the hemocyte population from these infected animals at 24 hours after they were infected. So then this is just a picture from our technician extracting the hemolymph, and that the hemolymph contains those hemocytes. And then we would image the hemocytes by confocal microscopy. So a couple things is we used a fly strain that has a GFP reporter in the hemocytes. So all hemocytes have GFP in them, and we also used a Coxiella strain that is tagged with M. cherry. So we would do 3D reconstruction, and we could cross-section these hemocytes. Again, the hemocytes are GFP, Coxiella has the M. cherry tag, and then we would also stain the nucleus with DAPI. So we can see Coxiella burnetii intracellular in these hemocytes, and we did a 3D reconstruction model to show, and we would take cross sections to show the bacterium inside the cell. So here's a video, so that the hemocyte spinning. Then we could take these cross sections, and we can see the Coxiella, M. cherry labeled Coxiella inside of that cell. So then here it again. So that's the whole 3D model. Take the cross section, we can see the M. cherry labeled Coxiella. So I mentioned earlier that the toll and IMD pathways respond to bacterial infections. So again, if you remember, Coxiella activates drosophila here, and this is mediated by the peptidoglycan receptor protein, which activates molecules called DREAD, FAD, and IMD, or immune deficient, and will activate an NF-kappa B molecule called relish that causes the induction of these antimicrobial peptides. On the other hand, the toll pathway responds to fungal infections or gram-positive bacterial infections, has a number of adapter molecules that will then cause the activation of transcription factors, mostly dorsal and dorsal immunity factor, and this causes the induction of defensin. So if you remember back to a couple slides, the toll defensin was not induced during Coxiella infection, but drosophila was induced. So we found in our Coxiella model that the loss of relish, but not PGRP-LC, would result in increased bacterial replication and reduced AMP induction. Again, just to go back to the pathway, so we're now knocking out either IMD or PGRP, PGRP or relish in the fruit flies, and then looking at how Coxiella replicates. So here looking at, so both PGRP and relish mutant flies succumb faster to to Coxiella infection, but only the relish mutant flies show increased levels of bacterial replication as compared to wild-type W1118 flies. And then we looked at Drosophila, that antimicrobial peptide readout, and observed that the relish mutant flies, they don't induce Drosophila at all, but even these PGRP mutant flies still show levels of Drosophila induction. So then we hypothesized that once Coxiella is replicating, PGRP-LC signaling is less active, and the infection instead activates relish for subsequent AMP induction through an alternative mechanism. And it's this alternative mechanism that we're pursuing, and we think that that it's replicating Coxiella generates an immune response mediated by an intracellular receptor, and we're hypothesizing that intracellular receptor is the STING molecule. So then, just to take us back to this pathway, we think that that molecule called STING is activating somewhere in this pathway that's further downstream of peptidoglycan receptor signaling, but upstream of relish signaling. So we're looking at, how does it activate immunity at this adapter level. So Drosophila encodes a STING ortholog. So this is a clustal alignment of STING from a number of diverse species, and we can see good alignment. Here's Drosophila. Second from the bottom, we can see the Bomix mori, and then here's mouse and human. And I'm highlighting here in red the cyclic dinucleotide binding domains. So these are the domains that bind the molecule that activates STING, the cyclic di-GMP, cyclic di-AMP, and you can see in all of the alignments, you can see very high levels of conservation at the cyclic dinucleotide binding domains. And here, zooming in, so here's cyclic di-GMP, and with human STING, it's been the protein's been crystallized, co-crystallized with cyclic dinucleotide, and it's been shown exactly which amino acids bind to which molecules or interact with which molecules of cyclic di-GMP. If we look at those amino acid residues in Drosophila STING, you can see very good conservation and identity in some cases of these exact residues in Drosophila STING as compared to human STING. So we believe that this Drosophila homolog is indeed the bona fide homolog of the mammalian STING. So again, these experiments are carried out by a grad student in the lab, Morena, and an undergrad, Marina, in the lab. So we found that Drosophila STING can bind to cyclic di-GMP. So this is doing an in vitro translation assay where we in vitro translate human or Drosophila STING or truncations. So this is just the N terminus of Drosophila STING 1 to 133, or the C terminal that contains those cyclic di-GMP binding domains. And we can see that we can pull down cyclic di-GMP with human STING, Drosophila STING, not the N terminal, but the C terminal portion that contains the binding domains. We can see interaction with cyclic di-GMP. If we make some deletion mutants where we delete certain cyclic dinucleotide binding regions, 1, 2, or 3. When we delete 1 and 3, we get reduced binding activity, but if we delete 2, we can still see decent binding with cyclic di-GMP. And also, these molecules are able to activate NF-kappa B. So the Drosophila version of STING can activate human NF-kappa B. So this is the full-length protein. So we can see very high induction of NF-kappa B. If we delete cyclic dinucleotide domain 1 and 3, we get reduced NF-kappa B activity. If we delete 2, we get slightly reduced activity, but not as significant as compared to the deletion of 1 and 3. So cyclic di-GMP does indeed induce a Drosophila STING-mediated immune response. So what we did here is we knocked out sting in Drosophila S2 cells, and then either mock treated them or transfected them with cyclic di-GMP. And you can see that when we knock out sting, we can see these genes are not as highly induced as when we use a scrambled double-strand RNA. So this is a normal immune response. And you can see some of these antimicrobial peptides that are being induced, such as atacin. And then if we categorize these genes into by gene ontology into certain categories, we can see that. So scrambled RNAi, again, containing sting, we see induction of these gene pathways, defense response to bacterium, antibacterial humoral response, defense response. But in cells that are knocked out for sting, you can see there aren't any red bars here because none of these gene categories are significantly upregulated. Drosophila sting, again, retains the ability to activate NF-kappa B. So now we're looking at mouse embryonic fibroblasts that have been transformed with human sting or Drosophila sting, then treated with cyclic di-GMP. So we can see in both that we can see translocation of NF-kappa B that is mediated by both human and Drosophila sting, but we don't see that Drosophila sting can mediate an IRF3 pathway. So again, the point is that human sting can activate both NF-kappa B and IRF3, but the Drosophila homolog only activates NF-kappa B and has lost the ability to activate IRF3. And we verified this by Western blotting. So again, these same cells, when treated with cyclic dinucleotide, we can see phosphorylation of I-kappa B, the inhibitor of NF-kappa B. So when this becomes phosphorylated, I-kappa B gets degraded and allows NF-kappa B to translocate to the nucleus. But cyclic dinucleotide was only able to activate the phosphorylation of IRF3 via human sting, not via Drosophila sting. So we next asked, how does Drosophila sting regulate relish during infection? So relish is that Drosophila homolog of NF-kappa B. So how, based on the results we have here saying that sting can activate NF-kappa B, how does it activate relish during infection? So what we did is we made a Drosophila overexpressing fly that we infected with listeria. And listeria is a pathogen that's been well known and well characterized to secrete cyclic di-GMP. So in our Drosophila sting overexpressing flies, we see increased activation of relish. And that's characterized by increased levels of this 50 kilodalton, 49 kilodalton processed piece of relish. So the C terminal gets cleaved and is retained in the nucleus, while the other portion can translocate. But by Western blotting, we can see increased levels of relish activity, which then activates these antimicrobial peptides, secrepin A2. So again, in our sting overexpressing fly, we get increased levels of secrepin. And again, that's mediated by relish, but drosomycin, which is mediated by the toll pathway. We do see some increased induction in the Drosophila sting overexpressing fly, but it's not as significant as secrepin. So the next question is, where in the IMD pathway does sting signaling occur? So what we did here is we used our Drosophila sting overexpressing fly, and we put that gene onto other flies that were able to contain RNAi for parts of this pathway. So we made in the same fly the ability of sting overexpression at the same time that there could be RNAi knockout of these genes. So again, the part of the toll pathway and part of the IMD pathway. So we did it, so we have all of our, so UAS DM sting means we're driving Drosophila sting overexpression, and at the same time we're knocking out parts of this pathway. Luciferase RNA, so that's just non-specific RNAi, and then we infected those flies with listeria, and we can see that secrepin, that IMD mediator. So when we overexpress sting and knock out either relish or IMD, we get the loss of secrepin induction. But overall, the overexpression of sting, we can see that there's an increase in antimicrobial peptide induction during listeria infection as compared to when the lack of Drosophila sting overexpression. And And then looking at drosomycin, that toll pathway reporter, we can see that there's really no loss in drosomycin induction during the overexpression of STING and knockdown of these pathways. We do see some loss when relish is knocked out, but there's a lot of crosstalk between these two pathways. And again, relish, a major mediator of the innate immune response, we expected that the loss of relish could impact drosomycin induction a bit. So the final steps that we're taking forward is to exploit the STING pathway to potentially inhibit Coxiella infection. So going back to Coxiella, there's really no good therapy for infection. People who are infected with Coxiella, it's a chronic infection, and the only way to treat infection is by years of antibiotic treatment. And in animals, there's no vaccine, there's no good therapeutic treatment, and the only way really to deal with a Coxiella infected animal herd is to kill the animals. So we want to think about other ways to impact or help treat Coxiella infections, again, by exploiting the STING pathway. So first we looked at levels of Coxiella replication, and we found that in mouse embryonic fibroblasts lacking STING, we saw increased levels of Coxiella infection as compared to wild type fibroblasts. And then looking at, so this is GFP labeled Coxiella, we can see increased amount of GFP signal in larger Coxiella containing vacuoles in the STING knockout fibroblasts as compared to wild type. And then what we did is we tried to activate STING before infection using cyclic dinucleotides. So if we primed and if we treated cells with cyclic dinucleotide and then infected them with Coxiella, we saw that there was decreased levels of Coxiella replication in primed cells as compared to wild type cells. Ultimately, we want to, so this is looking at the mouse system of Coxiella in STING, but we want to use Drosophila as a model for other insect vectors of Coxiella infection. So in addition to ruminant animals as being a major source of Coxiella, Coxiella is found in tick populations. And here we're aligning the STING molecule in humans, Drosophila, and the tick genome, looking at these cyclic dinucleotide binding domains. And again, we can see pretty good similarity in these binding domains, especially 2 and 3, between human and tick STING. Now, of course, the tick is not a good animal model to study infection, but if we can use Drosophila as another insect, that's a way to take a look into how potentially insects modulate Coxiella infection. So again, looking overall, we want to look at Coxiella infection, which produces these bacterial cyclic dinucleotides to activate STING. We want to see how this pathway becomes activated, and then can we use exogenous cyclic dinucleotides to activate STING and ultimately potentially decrease Coxiella infection. So again, delineate the role of STING during infection, dissect this pathway, and then prime the host immune response with cyclic dinucleotides to thwart infection. So again, coming back to the overall goal of the lab is to use multiple diverse species to uncover evolutionarily conserved host factors that are critical to the host response to infection. And with that, this is my acknowledgement slide showing everyone in the lab at a recent outing at a Japanese restaurant. If there's time, I can take questions from the audience. Thank you.
Sure. Thank you. Would you like to
Yes. Okay.
So let's start with the presentation and hearing one. Okay. Let me see. Okay. So my first question is, you talked about Drosophila STING and how it activates in human cells or cells?
Sorry, there's a lot of, I couldn't really hear the question. You're the activation of STING in human cells versus.
So you talk about the STING. It activates factor B. Does it activate beta in Drosophila or in human cells?
Yeah, so the NF-kappa B homolog in Drosophila is called relish.
Okay.
And Sting, the Drosophila version of Sting activates relish, and the human version of Sting activates NF-kappa B. But they're the same. They're, again, almost identical molecules. They both translocate to the nucleus and then activate the induction of innate immune genes.
Okay. The next question I have is that Coxiella, it is extremely pathogenic bacteria disease. What makes it so pathogenic?
Oh, okay. So what makes it so pathogenic is the fact that it can, when it infects a cell, it infects the cell in this slow metabolic form. So the small cell variant, that infection then goes into a vacuole, and then the bacteria actually replicates and becomes metabolically active in a protected vacuole within the cell. And inside that protected vacuole, the bacteria is actually in a different compartment from the innate immune response. So in that vacuole, it can replicate, and it's actually a very acidic vacuole, and it can replicate, and that it actually steals the metabolic energy of the cell. So animals and humans who are chronically infected with Coxiella, one of the major symptoms is fatigue, because the bacteria is essentially taking all that metabolic energy, replicating in this protected niche in the cell that doesn't see some of these innate immune mediators. And we think that the cyclic dinucleotides could be escaping from that vacuole and activating Sting, but then again, Sting can't, the results of Sting activation aren't able to reduce an infectious load inside that vacuole. But it's as little as one bacteria can get in and start replicating in the vacuole and can cause a chronic infection.
Wow, that's it. So you talked about cyclic dinucleotide and it has to go bigger animal. Some of the challenges.
Yeah, so exactly, I think the delivery, so the idea of using, you're referring to the idea of using cyclic dinucleotides as a therapeutic?
Yes.
Is that correct?
Yes.
Yeah, so I think the biggest challenge would be delivery of the molecule, and then delivery of the molecule in a form that wouldn't cause hyperactivation. So again, going back to the very beginning of my presentation, talking about the ability of if there's hyperactivation of an innate immune response in the absence of infection, you can get sort of this autoimmune disease. So we wouldn't want to deliver the therapeutic to cause, some sort of adverse hyperactivation of innate immunity, but we need to deliver it in a way that it would be less potent until an infection occurs.
Okay. Yeah. Information. So I have one. IIV is actually for V. And so you talked about horse as animal. Why not some rat? Why horse?
It's a good question. So West Nile infects dogs, cats, and horses, some of the larger mammalian models. I guess we just talked to, the reason I brought up horses is because our vet school is actually one of the diagnostic labs for West Nile infection in horses in essentially all of the Pacific Northwest, including Washington, Idaho, Montana. So this is a diagnostic lab where a lot of infected horses are seen. So there was I think last August, 2 horses came to our unit. So we were able to, the vets were able to deal with them. I guess infected cats and dogs aren't brought here so often.
Right. So one question because considering using IIV6 as a vaccine, but if you are getting things that are already, how are they going model?
Yeah, I think, moving that, some of those studies to any kind of clinical application would be more difficult. That was actually one of the discussion points is, we wouldn't, I guess it would be difficult to say, oh, let's just put a bunch of IAV6 in these mammalian models, especially if they come in after they're infected. Yeah, I don't think that would probably work very well. But again, coming back to one of our ideas of like priming the immune system, is that a way to reduce the load? But again, if we know where those horses were infected or any of those animals were infected, so where those mosquitoes are circulating, potentially we could treat the the yet to be infected horses as part of a herd on the farm, as an example.
Great. Thank for sharing information with us today. Really appreciate your time and effort, and thanks a lot.
Thank you.
Thank you. Goodbye.
Bye.